Okay, just making sure. And we should officially be live. Live from lockdown, live from the comfort of our own home. I just want to make sure and get confirmation that we are live. And then as soon as we get that, we can get rolling. Cool. We're good to go.
Okay.
It's like you're looking at me like you're like satisfied that it's worked. No. Welcome guys, welcome back to Barely Explained. This is a special live episode. It's the first time we've ever done a live episode, so I sincerely hope everything's working. The live chat is right there, so if I'm pointing correctly, it should be next to you if you're on a computer. If you're on a phone and holding it vertically, then it should be right below you. And then if you're full screen on a phone, it should be somewhere in front of my dad's face, I think. So welcome once again. We are doing a live coronavirus Q&A today. So we're back once again with Professor Burtram Fielding from the University of the Western Cape. He's the Director of Research Development, and he is also the Principal Investigator of the Molecular Biology and Virology Research Lab at UWC. He's been studying coronaviruses way back when, since 2003. So he knows what he's talking about. He's doing his stuff. If you look in the description below as well, you should see a whole blurb that I wrote out because there's a lot of titles to put there. So thank you for coming back.
No, thank you, and welcome to the audience as well.
Yeah, and not that you had a choice to come back because...
Not that I had a choice because we're in the same house.
Yes.
So I was pretty much forced to be here as well.
So as always, and how it's come to be with Barely Explained, let's just start off with some context surrounding this whole thing. As of the latest WHO situation report, so that's the one from the 9th of April 2020, we're sitting at 1,436,198 confirmed cases worldwide, and then we're sitting at 85,522 deaths. And then according to the NICD stats, which is local for South Africa, as of the 10th of April 2020, we're sitting at 2,003 confirmed cases. And that was derived from 73,028 tests conducted, and we're sitting at 24 deaths now. And also, of course, the latest news and probably the biggest and most annoying thing that's come out of this is that lockdown has been extended for 2 weeks. So if my calculations are correct, and I added 7 twice correctly, that should be to the 30th of April.
It does.
As we're going along, because this is live, if you guys have questions or you have anything that needs to be answered relating to the coronavirus specifically and the virus, then please put it into chat. We have people trying to collate all the questions, and we're going to try and address as many as we can in the time that we have tonight. So without further ado, the first question, just to open it up, is that the lockdown's been extended by 2 weeks. What exactly is the government trying to do by extending it? And then also, is it a good idea?
So it's always very difficult to give an answer when I'm asked about the lockdown. I understand why the government's doing it, and it's pretty much, as we discussed in the last discussion we had, it's pretty much about flattening the curve, as it's called, and it's really about spreading the infection curve, the number of those infected over a longer period. And as we mentioned the last time, it is about securing and making sure that our medical infrastructure can handle the number of severe cases. I do have some concerns about the lockdown, however, and maybe two major concerns, because this is not really a hard lockdown, because people can still go out. So that's one concern. So yes, we can have people not going out for most of the day, but I've noticed when people go out to the malls, for instance, you have limited numbers allowed into a shop, but people queuing outside.
And those lines.
And the lines.
Get very long.
So that creates a huge possible place for spread of virus. And then the second concern I have about this lockdown is you can separate people. However, without contact tracing, how do you track who has been in contact with somebody else?
So contact tracing just being that you need to figure out who that person touched,
Exactly.
who that person was near.
Exactly. And so if somebody becomes infected in Checkers, or somebody who is infected, is confirmed infected, visited Checkers, and they stood in a line with somebody, anybody in about a 2-meter radius could potentially have become infected. Those people go back to their homes. Everybody in those homes could be infected. They go out, they could infect others. So the problem with this almost soft locks that we're having is without contact tracing, how do we control this virus better, the spread of it? And I think that's my two biggest concerns. If we have a shutdown, but not all communities are shut down, there's a risk of not stopping this virus. If people go to collect the social grants,
and those are the old, elderly, disabled.
Exactly, and you have thousands of them queuing outside, the risk is there. And I think that's really my problem and my concern. And then another concern is if this virus behaves anything like the other 4 circulating coronaviruses. Remember, I mentioned the last time that we have 7 human coronaviruses. The 3 deadly ones are SARS, SARS-2 that causes COVID-19, the disease, and then MERS, and then we have 4 others that circulate throughout the human population on an annual basis that cause the common cold. If this virus behaves similar to those, where it keeps on circulating, so it will disappear, come back, disappear, come back, are we going to have this lockdown forever?
Yeah.
So that's another concern that I have. We don't know whether this virus can reinfect somebody after they've been cured, or whether after they've been cleared. And there's some data, I don't think that data is convincing yet. It wasn't done very well, those studies. But we don't know yet.
Yeah.
So that's my concern really with the lockdown and the way we're doing it at the moment.
So would you say it's almost too early to see if it's working? Because if we have a look at the size of our population, the population density we have in certain areas, 2003 confirmed cases isn't that much. Sure.
So it's 2,000, just over 2,000 out of 53 million people.
Yeah.
I think the numbers are actually much higher.
Naturally, because you can't test everyone.
Because we're not testing everyone. There is a rolling testing program at the moment. I think they said 10,000. Last figures I read, Germany was testing almost 500,000, 50,000 people a week. 500,000 over the short period. Then we will have a sense of who is infected and who to quarantine. Remember, this virus can be infected. Some new data shows you can be infected 2 days before symptoms appear, and then about 2 days, maybe slightly longer, after symptoms disappear. Contact tracing.
Yeah. And if we're not doing that.
And then testing. If we are not testing more people, then we might be missing it.
So would you say that the lockdown's a best option, or like is it? So first off, is there a better option that we maybe see in history? I know like there's nothing quite like this, especially in South African setting. And then also is it almost like the best of the worst systems in a way, like making the best out of a terrible situation?
I think based on the data that we have, it is probably a wise decision. But the flaws in the system, I think that can really come back and bite us. Remember that with SARS in 2003, quarantine worked really well. But the difference with SARS was somebody became really infective once they showed symptoms. So somebody would have a fever, then they could be isolated. Yeah, a person can be infected before they show symptoms.
Yeah, and some people don't show symptoms.
Some people don't show symptoms at all. And that's why I'm saying, if we do larger scale testing, I would not be surprised if the numbers of those infected are much, much higher. But they've just ended up with common colds or with no symptoms whatsoever.
So being diagnosed with pneumonia or common cold instead of...
People only go to hospital when they're really ill.
Yes.
Yes, we have tested. How many have we tested in South Africa now?
That was
5,000, I think?
Tested?
Yes, tested.
73,000.
73,000 tested.
Yeah.
How many of those were positive?
2003.
Okay, 2003 out of 73,000 tested. So it was those who were probably ill enough to go to hospital, and that's why they were tested.
Or the people who were concerned enough.
Or those people who were concerned enough because they've heard that if you're predisposed and you have certain risk factors that you need to be tested. Or they were in contact with somebody who could have been positive. So this is probably a good solution. But I think there's some, and that's just based on my opinion, based on my history as a coronavirology researcher. So I'm not an epidemiologist, and they would probably tell you something else.
Okay, and then just like looking into it, do you think there could be a chance if maybe this lockdown gets made shorter, that we don't use the full 2 weeks, or is it pretty much set in stone? We have to let it run its course. One person in chat suggested rolling lockdowns, maybe like they have rolling blackouts.
So rolling lockdowns would probably not work because you would end up with the same type of problems. I'm still saying that we should target those who are higher risk, so the elderly and medical personnel. Yes, I know that if you look at stats in other countries at the moment, that it's not just the elderly that's developing severe diseases, but if you look at those stats, it's still predominantly male. And even in New York, I think there was a very large population of African Americans that were infected with, some of them, quite a large portion, very severe symptoms. But in that population, there was a very high incidence of heart disease.
Interesting.
So it's the underlying condition. So if we know who are at greater risk, we should, in my opinion, target those, I think, better and protect them more.
And then many opponents of the lockdown would say that perhaps we're doing it too early. Do you think that this could be extended again if they don't see the numbers that they want?
So to be honest with you, I have absolutely no idea. I was totally shocked yesterday when the president made the announcement yesterday. So I'm not involved with any of the task teams, the ministerial presidential task teams. So I was shocked. I expected him to wait on more figures and more data, and I thought that there would probably come an announcement sometime next week. So I was, I think, just as shocked as everybody else. There were some rumors going around in certain circles that I'm in, but I thought those were just rumors, as with anything else. So these decisions are not taken lightly. These decisions are based on expert input, on numbers, and on predictions. So it is predicting how this virus will react. This virus has taught me one thing: it's not behaving close to the way we're thinking and predicting that it should be behaving.
Okay, and then we've been speaking a lot about testing and testing enough. Just so, like, to give people an idea of what does the testing look like? What are they testing? What swabs are they taking? And what are they doing it behind the closed doors in the lab?
Okay, so it is actually not that complicated. And I'm speaking as a molecular biologist.
Okay, yeah, of course.
So
with a PhD.
With a PhD. So if you ever watch CSI, if they go to the crime scene and they collect DNA from the suspect, they then go back to the lab and they do this really cool thing where they have a test tube and they have liquid in there and it shakes, and then they take it out and then they run it on this gel and they view it under, and something glows and they say, oh, I can see something. So this is very much like CSI. So pretty much when you are tested, they would take something that looks like a very long earbud. So this thing is sterile.
Okay.
This earbud will be, it's called a swab. This will be sterile. They will take a sample from your nostrils and they will take a sample from the back of your throat.
What do you mean by take?
So they stick it, it's a very thin stick with a cotton swab in front. So they will stick in there, because remember how's this virus spread? Respiratory samples from the nose and from the mouth. And then you can transfer to the mouth, the nose and the eyes. And that's why we're telling you to keep your hands clean. So once they have this, it will go back into this sterile tube and it will then be taken to a lab. What the scientists would do over there then becomes very CSI. They then take this, because now remember, you have some of your body fluids on there, and in your body fluids you have the virus if you're positive. They will then have various techniques to take the genetic material out of that virus. Now remember, genetic material, we all have genetic material. It determines your race, your height, your hair color, what type of hair, your color. That's genetic material. So they will then remove the genetic material from the virus, and then they will use very specific molecular biology tools to see whether the virus is present or not. If it is present, they would then be able to see it under a specific UV light, or under a computer that reads the sample. So you would then see a positive result.
Like obviously looking for those markers.
Something could glow.
If
they do it very large scale, something could glow. If there's no glow, then the virus was not present. If it does glow, then the virus was present. So various labs could use different techniques, because we have various tests now. And that would pretty much tell you whether you're positive or not. So there is no truth to the rumor that these swabs are contaminated with anything, they have to be sterile.
they have to.
And in fact, if anybody comes to you as part of the government's testing program, they need to break that seal in front of you. The person needs to wear gloves. The person needs to be covered. To protect both yourself and the person. Any swab that's not sealed could potentially be infected, could be contaminated, which means that you could come back as a false positive.
Yes, and it could infect you.
Well, not just infect you, it could infect you, but also now a test come back and says that you're positive. But you're not positive, now they put you into quarantine, not just lockdown. So make sure that those seals are on it has a little seal that if you just twist it, it breaks. Just make sure those are in contact. But it's very CSI type of vibe to the whole thing.
So I understand that in Singapore you worked on a rapid testing kit. How close are we to one that's viable here in South Africa?
So I've seen rapid tests. I know South Africa is working on it as well. 2 labs in Singapore have developed. I think 1 or 2 labs in Europe has developed as well. That works slightly different. So there you can get the result between 5 and 15, 20 minutes.
So that's like you just sit in your car, you wait for the result.
So the doctor would essentially take a blood sample and then put the blood sample on something that looks like a pregnancy test. The pregnancy test, so this test, would have pieces of the virus on there. So the virus is broken up and it's put on this test. Your blood contains what if you've been exposed to antibodies. These antibodies will recognize the proteins of the virus on that test.
So it's like it's fighting the virus on the test. And
No, so it goes and it attaches because it's very specific. There's an affinity, so the one will recognize the other. If you then have this binding, then you have a color change, exactly like a pregnancy test. So within 5 to 20, 25 minutes, if you have a line or you have a plus, it means that you're positive. So if nothing, then you're negative.
We're going to see this being rolled out because
I'm sure because it is not these tests are not as sensitive as the genetic tests. But it's a very good initial screen and slightly cheaper because now immediately somebody can be quarantined while they're confirmed. So it's not a confirmatory test, but it's a very good initial screening test.
So just keeping it to lab work, there's another question that came through in Afrikaans. I'm going to read it in English. There was a breakthrough that was made at UWC surrounding the virus recently. I think it was with Sandy. Could you explain what that's about? Because it's South Africans actually doing something. But what did they do?
So there's a few South African labs actually doing very, very good work. This work is from the South African National Bioinformatics Institute. And bioinformatics is essentially computer gurus who take genetic or biological information and then analyze it. So what they then essentially did, they worked with another 1 or 2 other groups in South Africa who then isolated the genetic material from samples taken from a patient. They then determined what the sequence of the genetic material is. Sandy then took that material and analyzed it and put it together to show this is what the genetic material of the virus looks like. What does that mean now? So now we know,
sorting it into a library type thing.
Exactly. So now we know that this virus at the genetic level looks like this. We could then analyze that and say, hang on, maybe this drug would work the best. Hang on, maybe this vaccine would work the best. So it is understanding the virus at the level that determines what that virus's role is and what that virus can do and what it will look like.
Okay, so there's some very exciting stuff. It's cool that we're seeing it and that's making waves. So then with that, with knowing more about the viruses, there's been rumors that certain vaccines are believed to help stop the spread. So I believe there was rumors about a TB one, there was a few others that help stop the spread of this virus. How true are these claims?
So those are not vaccines, those are antivirals, antiviral drugs. Remember, antibiotics cannot be used to treat viruses because viruses are not alive. So you need antivirals. So antivirals typically either stop the virus from making more virus by interfering with the genetic material, or it could even prevent the virus from getting into the human cell once it's gone into your respiratory tract. So the vaccines is something that you inject. Remember, as a child, you get BCG and you get those type of vaccines.
There were some rumors about BCG as well.
So we'll get to that now, because that's a very interesting and very important one to touch on as well. So I've read reports now where 2 vaccines are being tested. Any new vaccine that will be tested specifically for this virus needs to go through the proper processes. And one of the important first steps in a clinical trial is to show in healthy people that it will not cause any adverse symptoms. So it needs to be tested, then a bigger population, then a bigger population, then a big, and then can be rolled out. So this is only stage 1, phase 1 of clinical trials.
Okay.
There's some very promising data that some of the drugs that we're using for TB, some of it we're using for malaria, some of it we're using for other viruses, seem to be working against this virus.
Which is interesting that like drugs that work for bacteria like TB are working.
So it depends on whether do those drugs interfere with the genetic replication.
Okay.
If it could, then yes.
Okay.
But that's why I'm very cautious when it comes to this, because you read various reports from various parts of the world, and there's conflicting data. So until it's reproduced on a very large scale consistently, we cannot claim to have found a cure.
So don't believe everything you read.
Don't believe
take this drug, take that drug.
What you need to do, you need to leave it to the professionals out there, to the medical professionals who would go and would vet information based on what the medical councils tell them, but also looking at the publications. Then you mentioned that there are reports that all kids in South Africa are immunized with BCG, so it's mumps, rubella, and another disease, and so we all get it. So there's some anecdotal evidence, so it appears the data, when we look at the data, we're kind of seeing this, that in countries where kids have been immunized, so as adults you still have the antibodies, they were not developing the severe symptoms. So there is some data that's saying, hey, hang on, maybe there's some cross-reactivity, because remember, typically a vaccine protects you only against a particular one virus. It looks as though there's some cross-reactivity. Even for this, I would not become excited yet.
Okay.
We need to look at more studies, bigger studies, and more confirmed peer-reviewed studies. However, a colleague of mine that I'm on a group with, he mentioned this about a month and a half ago, that in Iran, they were seeing exactly the same thing. So the Iranians, as far as I know, is looking at this.
Okay.
And remember, they have a very big population of infected. So when it comes from them, that study would tell us a bit more.
So at the end of the day, trust sound science, always cross-check everything. You can't just expect a WhatsApp that comes in.
Exactly.
And then on the subject of vaccines, how close are we to actually having a vaccine for this virus specifically, if there's any progress that's been made?
So if they're doing phase 1 trials now for two vaccines, others are a bit later. I've seen where scientists, researchers have predicted best case scenario maybe August, September next year. Because remember, vaccine needs to be safe, vaccine needs to be efficient, and it needs to be specific preferably, and it must not amplify symptoms.
Okay, because it could very well do that.
It could, if it's not tested properly, and it needs to be safe. The vaccine cannot make you sicker than what the disease would make you. And that's why vaccines normally take a very long time. So if we have, and if labs have the proper testing and quality assurance protocols, which they should have, then probably middle next year, third quarter of next year.
So do you see a possibility of us having to lock down again until we get a vaccine?
I certainly do not hope so. Remember, vaccine does not cure you. A vaccine protects you.
So it's not like you have it, we can give you a vaccine and you cure. It's not a cure.
No. So it would protect you from a subsequent infection, and it will prevent that infection, if you do get it, to not become a severe infection again.
Okay.
And much milder. So you're still exposed to mumps and to chickenpox, but you're protected because your body has an immune memory, and as soon as it sees this virus, mumps and measles and German measles, then it protects you, because the body can jump into action and can protect you. So it's not a cure, it is a protection. The antiviral drugs and other drugs that we use that are being investigated, those are cures.
Okay.
So that's what I think. That's what I am more excited about. Because even with a vaccine, if you want to develop herd immunity, you need about 60% in South Africa being vaccinated before you really see a result. Then also the question: will this be a lifelong vaccine? Because not all vaccines are lifelong. Do we get boosters? How often do we get boosters? Will this virus come back every year? Does it circulate? In the end, we don't know those things. Those are all questions that we need to answer.
And then surrounding that, the chances of this virus possibly mutating so that people who've gotten it can get it again?
So there are some studies already out showing this virus is mutating, but it is very small mutations. I don't think it will become deadlier. The mutations are normally very small mutations. Because remember, viruses, in our first talk we had, we discussed viruses co-evolving with their hosts. So a virus found in a bat, and if it's a natural host, the bat, the virus does not even create symptoms in that bat anymore.
Oh, it's just so used to the system.
So the two adapt to each other to such a degree that viruses are not out to kill people. A virus needs a host to make more of itself.
Yeah.
So a good virus needs to keep its host alive as long as possible with as low immune system actioning as possible so that it can make more of itself. So there will always be this co-evolution. So if we take this virus, any other virus that grown in the lab, you can actually see after a month or two continuous growing, the virus that we have in the end is not the same as the one that we started with.
Okay.
So the virus changes to its environment that it's in, so that it can find the optimum virus to grow optimally in that environment.
So mutation is almost a given in this regard.
Oh yes, definitely. It's an RNA virus. So RNA viruses mutate. So mutations will happen. So can I just answer your question further? So the question is not will it mutate to come back? It is how does the body's immune system remember, how well it remembers this virus, so when it comes back, that we still recognize it. Because the body's immune system might need another boost to say, hey, you need to look out for this thing. Don't forget about it.
So
we don't know that.
Like the seasonal flu almost.
Almost like, but remember, seasonal flu is a bit different because seasonal flu has what is called antigenic shift and antigenic drift. These are very small and big changes in the viral genetic material, and the virus literally looks different next year.
Oh,
so it's a large portion of the genetic material that is substituted and changed. So it looks totally different, and so the flu for this is different. It will look different next year. And that's why our vaccines are different every year.
Okay. And then just moving it on to a slightly different thing, like with any major news thing, like that happens in the world, there's always conspiracy theories. And I know you enjoy reading up on conspiracy theories because there's a lot to read up on. But one of the big ones is the correlation between 5G and coronavirus. Are these claims, do they have any basis to stand on at all? Or is it just complete garbage?
So I've looked into this because I've had some students asking me this as well.
Okay. And these are medical bioscience?
These are medical biosciences students. So I looked at the various theories out there, and conspiracy theorists, and you're right, I love conspiracy theories. Conspiracy theorists will tell you they're conspiracy theorists because people are trying to hide the proper information. So I've seen a few. I've seen where the presenters just claims that there is no virus. 5G is making us ill, and what we're seeing is due to the 5G. I've seen where the speaker has said that 5G poisons the cells, and then the cell secretes genetic material and proteins, and that is exactly what the virus is. So it's not a virus, this is what it is not true. So I do not know whether 5G causes cancer. I do not know whether it does anything to the immune system. I do not know those things. I'm a coronavirologist, so I'm speaking from a coronavirology point of view. This disease has been shown to be a result of a viral infection in the human body, and the virus that infects, that causes COVID-19 is called SARS coronavirus 2. How do we know that? There's something called cost postulates. And about 150 years ago, this guy said, hang on, how do we know that a disease is caused by a particular bacterium? Remember, not viruses, not bacterium. He then said, so let's see, everybody that has the disease should have the bacterium. If I take out this bacterium and I purify it and I put it into a healthy host, that person should develop the same symptoms. Then I should go to be able to take this new person, take out the bacterium again, then I should see the same bacterium. Cost postulate. So what did I just show? This person has symptoms. I take out the bacterium that's causing those symptoms, put it into a healthy person. They develop the same symptoms. I can then take it out again. I've just shown that this thing that I've put into a healthy person causes the same. That works for bacterium. It does not work for all diseases.
Okay, yeah.
So there's people who would tell you, but cox postulate cannot hold for everything. I agree with that.
Okay, yeah.
Somebody then came along and said, but hang on, for viruses this doesn't work. So the guy Rivers came along, 1939, and he said, no, we need to modify cox postulates. So he then said, not everybody that has symptoms, because remember this is a syndrome. So it's a group of symptoms that you have together when you have an infection. You might not have all the symptoms. He then said, if you have some symptoms and some people, you go and you take out, you see this virus, you get it out, but what if the person does not have the virus anymore? What if the person has the genetic material of the virus in there? We can take out either one, put it into a new host, and we see the same thing. But we do not have, you would not volunteer to be part of a cox postulate experiment.
Yeah. No, you'd have to pay a lot of money for that. Yeah.
Nobody can pay me enough to volunteer. For HIV, you have to do exactly the same thing.
Okay.
Nobody would volunteer.
Yeah.
So what do they say? If you can't find a human host, so you can't use, you need to find an animal host. So 2 studies now have thus been that I've read that have shown that cox postulates have been fulfilled. So they've taken this SARS-2 virus that causes COVID-19 and they've put it into golden Syrian hamsters. The golden Syrian hamsters became ill with respiratory disease. They could spread it between them, and when they took it out and put it into the new host, they could become infected and you could get it out again. And then another group came and took normal mice, changed it so that the lungs of these mice became humanized. So they inserted ACE2, the receptor, and they did the same experiment. So there is fairly conclusive proof that COVID-19, what we call COVID-19, this syndrome, is caused by this virus that has been isolated. So not everybody might have the virus, not everybody who's infected will have the virus or the genetic material. Not all of us develop flu every year. Because it depends on the strength of the virus, whether the virus is competent. It depends on the host immune system. So not all, scientists would say, not all viral infections are productive. You can have an unproductive viral infection as well. So cox postulates for this, I've seen 2 papers now where it has been fulfilled.
So bottom line, like it's not 5G that's causing it. There's a virus that's causing the COVID-19. Okay.
And as a scientist, for me, that would be conclusive proof that it's exactly the same experiments that was done with SARS in 2003, where a group from the Netherlands and America, they did experiments together, and they conclusively used cox postulates as well to show that SARS is causing this disease, SARS coronavirus causing SARS.
Okay. So just a reminder that we are live. So please, if you have any questions surrounding COVID-19, the coronavirus, just stick it into the chat, which should be either next to me or right below me. And please, we'd be really happy to answer those questions. We have a few questions in chat, which I want to specifically ask, and we can start having a look at. So please send those questions through, because that's why we're here. It's a live Q&A. So the first question from Lyndon Benting is, are viruses and exosomes the same thing?
No, they're not.
They're not the same.
So a virus is a very simple non-life entity, and it essentially consists of a protein lipid layer surrounding genetic material. Exomes is part of genetic material in various other organisms. So they're not the same things. There's some theories that says if you look at where viruses come from, they could have originated from exomes and they became these separate entities. But that is just a theory, but they're not the same thing now.
Okay, cool. And then another question from Yaku Maret. It's a bit of a long one, so I'll try and paraphrase it a bit. But so essentially it says, it seems as though the spread of the virus is slow during the lockdown. If we are striving for herd immunity at 60% of the population, this implies that 36 million people out of 60 million South Africans catch the disease at some point. Seems quite far away, and each round of lockdown will push that further into the future. Assuming vaccines are 12 to 18 months away, should we just hunker down and wait until a solution arrives in the form of a vaccine? Or are there other strategies besides all-out lockdown that we can use?
I am very hopeful that we'll find a drug that works very soon, and maybe not a new drug, but a drug that's currently used for something else. And then I think a very important thing that we're missing is we need to protect our medical workers, because ultimately somebody who's very ill with this disease will be the most infective. So we need to protect them with protective clothing, and we need to, those who are at higher risk, those are the people that we really need to protect better. There's something that we're forgetting. If you look at the 4 circulating coronaviruses, typically they cause mild cold-like symptoms. When they do cause more severe symptoms, interestingly, they then found with other viruses. So there's a co-infection. We have not, as far as I know, we've not even looked at what type of co-infections are we getting. So your dog that you take every year is inoculated, vaccinated against a canine coronavirus. And it's a canine coronavirus that infects the intestines. And it's not lethal anymore. But if the animal does become infected with this canine coronavirus, it then makes the canine more susceptible to infection of another virus, a parvovirus. Then it becomes deadly. So interestingly, when you have this co-infection between this canine coronavirus and parvovirus, they become deadlier. Are we seeing the same? So we're seeing this for the other coronaviruses, more severe symptoms. Is it the same for this? I don't think anybody has looked for that yet. So we really need to look at that. And then just interestingly, canines also have a coronavirus that infects the lungs.
Okay.
So they have 2.
And then another question from David Howard. If we were faced with the need to extend lockdown beyond April, are there ways that certain industries can safely start operating again? What measures could be considered?
So for this virus, once again, I think if somebody is at higher risk, they should preferably be not isolated, but they should maybe observe the lockdown. And then the most effective way of spreading this is the good respiratory hygiene. Wash your hands. Cough into a tissue or your arm. Wash your hands as frequently as possible and as effectively as possible. Don't touch your nose, don't touch your mouth, don't touch your eyes. Often when I do an interview, do I touch my nose and my mouth?
Same here,
and my eyes. So that is the most effective way of preventing this. So that 1.8 meter social distancing that the WHO has said right from the start. Those are important things. Yes, this virus can be spread from the respiratory tract. There's new data that says it does not just spread from the larger droplets when you sneeze and cough, but also aerosols. So aerosol is a very fine spray, especially in medical settings. So aerosols have been shown to be a problem in medical settings, when they do a lavage, for instance. And that's how we discovered in Singapore why doctors were so at risk. When they would stick a tube down the person's throat to get samples from the lungs and the airways, it would create this aerosol plume, very high in viruses, but in closed, smaller spaces, that's a risk. So how do we stay safe? By practicing that good respiratory hygiene practices that everybody probably knows by now, because we've read and heard about it everywhere.
So even though this lockdown will inevitably end, we're all going back. There's a new normal which we need to live with.
Yes, and that is part of the thing. So if this becomes a virus that circulates yearly, so it moves between hemispheres, we can't stay in lockdown forever. So we need to change the way we live. We need to change our practices. And it's exactly the same. I ask myself, why are we doing this now and why are we preaching this now for this coronavirus, not for influenza? There's a very interesting study where this group looked at what would be the ultimate death rate for this virus. They're saying, yes, we're seeing now between 3%, 4%, in some cases 8%. But they're saying, remember, this is not a death rate. This is a case death rate. It's a case mortality rate. Their calculations predict that at the end of this, when we have tested as many people as possible, we would probably see the higher limit mortality rate of only 0.66%. That is vastly different between 0.66 and 4%. Influenza, seasonal influenza is 0.1, 0.2%. So yes, it's still 6 times higher, but it depends on the type of influenza. Avian influenza kills between 30 to 65, 70% of people infected. The only thing is it doesn't spread easily between people. It's mostly from chickens to people. Swine flu was higher than the seasonal flu. So if in the end this is the death rate is very similar to influenza, why not change our practices for all diseases? So good respiratory practices for in general would protect us from influenza probably as well.
So just once again, we are live. If you have a question you would like answered, just stick it into the chat. Professor Fielding, my dad, would be able to give it a shot and try and answer it. And then just one question that a lot of people's been making memes and jokes about it. This all started because someone ate a bat. But like taking it a bit more serious, like how serious of an issue do we see these exotic animal like wet markets where for like human consumption? Like how often do we see a jump from those type of environments?
So I think we miss many of these. So there's many that we will not know of. So if you looked at SARS in 2003, when they went back to the animal handlers at those wet markets, many of them seroconverted, which means they have produced antibodies at one point or the other. They were infected to this virus and they produced antibodies. They survived. So a year, 2, 3 months, we don't know, but they've been previously exposed. So I'm sure there's other viruses that jump as well. But because they're so mild, we will never know. Only a small fraction of respiratory diseases that medical staff and researchers believe is virus induced. We know the virus, the one causing it. The vast majority, we do not know what's causing the viral disease. So there are many viruses out there we don't know, that we've never isolated. The MERS coronavirus, when they went back and they tested the camel farmers, they were seroconverted, they had antibodies. If you look at Ebola, Ebola is linked to the trade of bushmeat. So the great apes. So there's a jump over there as well. So I think we will see this. Much of it I think we miss because it's just not severe. So we just do not see it. But there's many of these that will probably still come, and we just need to be as ready as possible.
Yeah. And then just going back to another question, we touched on this slightly earlier. Jenna McCullum asks, does Mr. Fielding think that there will be rolling lockdowns?
I hope not, but once again, I'm not part of any ministerial team or advisory group. If the clever people do their calculations and they think that it could help, then it would probably be instituted. I'm a bit concerned about that because our practices have not changed. Yes, we're in lockdown. Drive through certain areas and see that people are not maintaining the lockdown. So you can't have a portion of the population maintaining lockdown and then everybody queues somewhere a bit later. I said earlier, Singapore, it worked because with SARS-1, you were infective when you had symptoms. So you could physically remove that person from the population and keep him separated until he cleared. We're not doing that at the moment. Somebody could be infective and they could still stand in a queue somewhere and we will not know because we're not contact tracing. So I do not know. I guess the president will make that call in the end based on what his advisors tell him.
And another one, Peter Griffiths asks, your thoughts on the lockdown being lifted in the Wuhan province in China?
The Chinese government, obviously, similar to the South African government, make those decisions based on the data that they have. Interestingly, I'm actually being interviewed tomorrow by Chinese television, apparently, asking me on the same topic. At one point or the other, governments have to make that decision. They've been in lockdown for an extremely long time. I still think if you go now and you test, you will probably still have positive cases. And I can't say this enough. We need to identify who the at-risk people are, and those are the people that we really need to keep safe and target. So my advice would be, if you know of somebody who's at that higher risk, implement measures to try to keep them safe.
Yeah. So I think we have time for at least just the last few questions. One of them being, Tara now asks, is it you or your dad that is the babe magnet, as per the mug?
We must have that debate, I think.
Okay.
It's still not decided.
Okay.
But we can have a discussion afterwards, and I think your mom can vote.
That sounds a bit biased, but okay, thank you. And then Anne Watt asks, can animals get the virus? So I know we touched on it the last time around. So
this virus, I've seen some studies. So there's a very recent study that has said it's been detected in tigers. There are some studies that have said they've detected it in cats and in dogs. I've looked at the cat and dog ones, and I think it's one of those where we need to be very careful that it needs to be done on a larger scale. I looked at I think it was the dog and cat one, the particular one. So I'm not saying the tiger one was a bad study. The cat and dog one that I read was a very poor study. Essentially what they did was take a sample, place it into the animal's nose, and then a couple of days later they took samples from the animal's nose and from the lungs, and they detected virus in the animal's nose but not in the lungs.
Okay.
That's a bit dodgy, because if you insert something somewhere, you're going to get it over there. So we need to determine two things. Remember I mentioned about productive and unproductive and non-productive infections. So even though an animal could become infected, is it productive? Can it then infect others? It's very difficult, and that's why Koch's postulates take so long to be proven, because you need to find a susceptible host that has symptoms similar to what the person has or would have, and that is infective. And that's why it takes so long. So for people that will probably find this virus in animals, it does not mean that the virus can replicate, make more of itself in that, and then become infective. It does not mean that the virus is actually in the airway. It could mean something as simple as the test has to be repeated, and it needs to be repeated on a larger scale. So some of the data appears as though it can. There is no data that shows that it can move from the animal back to the humans. There's very little data, not conclusively, to show it can move between animals, tiger to tiger. I'm not convinced yet, but I would not be surprised, but I would be more surprised if it could then move back to the humans.
Okay.
And yes, if it gets into the animal, it's probably from you infecting the animal, but it does not necessarily mean it can get into the animal's airways and its cells, get into the lungs, replicate, and then become infective. That has not been conclusively shown yet.
And then I think we have time for one more question. This is from Morgan Morris. He asks, how is it that a human coronavirus can now infect animals? Isn't that just supposed to be one-way traffic, animals to humans? I know, like, you just touched on that now.
So it's called, viruses have tropism, and viruses typically have very narrow tropism, because tropism means the number of hosts that it can infect. And that's why I'm saying I would like to see more data. We do see, remember, bat to MERS, for instance, to camel, and then to human. And sometimes we could then, I think, see human to camel. But there you need receptors that is found in both. And receptors are proteins sticking out of your cells to which the virus can bind. If a cell in an animal does not have that protein sticking out, it cannot bind and get inside. So that needs to be proven. So the second study I mentioned, where these guys humanized mice, the lungs of mice, they had to insert the ACE genetic material into the mouse, and then the mouse lungs made H2, and that was on the surface of the cells of the mouse. They had to humanize the mice. So it's very unusual, this intertropism thing, or the very broad tropism. So I need to see more conclusive data that I haven't seen yet.
Okay, so that's pretty much all the time we have for today. Thank you so much for joining us on our first special live episode of Barely Explained. Please give a like and subscribe. If you want us to do this again, let us know in the comments, because then we can maybe make a bit of a thing out of this while we're all locked away in our own homes. Also, if you subscribe to our channel, we have other content that's not just heavy and coronavirus related. We do a podcast. There's Today in a Teacup. So please go check that out. Enjoy it. If you think your question wasn't answered, or you still have a question that needs to be answered, check out our first two videos. You'll find the links to those in the description, and then also you'll find it on the actual channel. And then also special thanks to Professor Fielding, my dad, for coming in all the way from home. And then also thank you to our chat moderators as well for feeding the questions through. So follow us on social media, let us know if you enjoyed it, and let us know if we should do this again. Okay, cool. Thanks, guys. Bye.
Bye.